Marfan syndrome is an example of heritable thoracic aortic aneurysm with dissection due to mutation in a matrix component. The matrix component is the molecule fibrillin 1, which is assembles into microfibers that can or cannot associate with elastic fibers. They have multiple functions, including modulating local TGF-beta bioavailability. And in the media of the aorta, they play an important function by serving as a molecular bridge between the elastic fiber and the smooth muscle cells, thus connecting what we can consider as the mechanosensing, mechanotransduction unit, contractile unit, in addition to regulate, as I said before, the bioavailability of latent TGF-beta complex. Two mouse models have been developed and mostly used for studying Marfan syndrome pathophysiology. One is the so-called fibrillin 1 C1039G+, and the other one is the fibrillin 1 MGR mouse. The first one develops aneurysm, but rarely dissect in a very long period of time, whereas the MGR mouse has a much faster development of disease and leads to dissection. In earlier study by Abashi, it was shown that blocking losartan or blocking TGF-beta had the same phenotypic outcome of rescuing the phenotype, thus suggesting that the activated AT1 receptor signaling was upstream of the TGF-beta, and the TGF-beta signaling hyperactivity was driving the disease. Later, Tim Baxter showed that the efficacy of losartan treatment wasn't as strong as in the previously shown in the model of the more severe model of Marfan syndrome. Here is just an experiment that shows how this experiment then went in our lab carried out to show that the treatment with the losartan did improve, as Tim had shown, the survival, the immediate survival of the MGR mice, but to our surprise, treatment with a neutralizing antibody actually killed the animal earlier. So this experiment refuted the idea that TGF-beta was downstream of immediately downstream of the AT1 receptor. And they also indicated that during the early phase of postnatal life, blocking TGF-beta was deleterious. Much of the study conducted by the lab of David Vicek and George Telides and also Mary Shepard have brought to this current understanding of the disease. Initially, impaired availability of TGF-beta, latent TGF-beta, leads to loss of protective TGF-beta signaling as a result of fibrillin 1 deficiency. And later on, as a result of increased wall stress, there is a stimulation of TGF-beta-driven matrix remodeling. This phase, and only this phase, appear to be the one sensitive to losartan, as demonstrated by Mary Shepard, who communicated this at the last GenTAC meeting. Many other studies by many other investigators have come out with a number of biological targets involved in driving the disease or compensating for the disease. And as you can see, the number is very high and the interconnection are unknown. To overcome this problem, we recently took a different approach, which is a system biology approach. In this approach, transcriptome are generated from arterial tissue from Marfan mice and Marfan patients and compare in order to find shared pathway associated with disease. I have already reported at the last meeting about the approach of drug repurposing, which has led to the identification of baclofen as a potential drug that can be used in combination to mitigate the progression of the disease. And this was published recently in JC Insight. Today, I will discuss about the pathway discovery, in which, again, we start from differentially expressed genes here shown in this volcano plot. From these genes, through computational analysis, one derives the binding site for transcription factors, which are then scored according to their statistical significance. And from the interactome, there are derived the protein kinases that are believed to modify the transcription factor that are in turn modifying the expression of the target genes. This work was done by Cristina Ciaresco, and she, in collaboration with the French group of Catherine Boulot, compare the transcriptome of the Marfan mice at P16, P30, and P60, and Marfan patients, obviously, endpoint post-surgery. The number of samples is interesting, but only 3, 3, 3, and 3. And even with this small number, we reached statistical significance. And across all of them, the protein kinase that was mostly represented were in common between the mouse and the human samples, and which selected IPK2. By the way, MAP kinase 14, which is ERK, showed that we had an internal control that was a valid one. IPK2 is being shown by Jean et al. To be a stress stimuli induced protein kinase that is involved in modulating a number of pathways, including TGF-beta and P53 in kidney fibrosis. Based on this data, we validate this data by showing that during progression of the disease, there is an incremental, there is a constant elevation of IPK2, here is in the protein, and this is qPCR. And more importantly, when we generated mice without IPK2 and crossed to our Marfan mice, the mice were show a significant mitigation of the median survival. And this was accompanied with mitigation of the aneurysm, as well as all the other histological criteria such as elastic fragmentation, collagen accumulation, and cellularity. Using the same computational approach, we also looked at predicted IPK2 substrate, and in the mouse and the Marfan mouse and patients, we found in common top P53 and the TGF-beta. There are others, but we selected that to validate the approach and show that genes like CTGF, which is downstream target of TGF-beta, are elevated in the Marfan aorta, and in the aorta of the Marfan mice without IPK2, that elevation is almost eliminated. Also, phosphosmad shows a reduction with the IPK2 deletion, as well as P53. We then decide to use a compound, B73, which has been shown to allosterically inhibit the IPK2 SMAD3 interaction. And this is a control to show that indeed we had that interaction. We go from Marfan to Marfan treated, there is a diminution of the SMAD3. And treatment with this compound increases the survival of the mice with the same characteristic that I showed you before in the genetic deletion. The same thing with elastic fragmentation, collagen accumulation, and cellularity. Perhaps there are some differences in terms of in terms of fragmentation is less effective, and also in terms of cellularity is less effective than the genetic deletion. Finally, these mice aorta were isolated and sent to J. Humphrey, who performed a biomechanic experiment, which showed some amelioration of the circumferential stress and the stiffness, but little or none of the elastic energy and distensibility. These results imply that the compound effectively mitigated the phenotype, but did not restore completely all the biomechanical property and therefore tissue functions. So to conclude, B173 is yet another drug that we have identified with another target in trying to generate eventually a protocol that use multiple drugs to target the different steps of the dissecting aneurysm progression. I will leave with the name of the people who have done the work, and thank you for your attention.